Thank you very much, and thank you very much for organizers. My biological clock actually went crazy. I came from Brisbane, Australia, where it's tomorrow morning, 4 o'clock, and we are in the middle of the summer, 36 degrees Celsius. And this gentleman here is my favorite picture. Normally, young generation don't know how a lecture theater looked like about 60 years ago, even when I studied about 30 years ago. This guy is Professor Ugo Schiff in Florence, Italy, my favorite picture. This is the university where I came from, which is in Brisbane, is the northeast part of Australia, as I said, in the middle of summer. And this is the most important slide of my talk. This is my research group, just happened in our recent group get-together. Obviously, all the work, what I'm presenting, came from their mind and very able hands. I work about 5, 6 postdocs at the moment, 13 PhD students, and different honor students. And I just realized, because I was asked, that I'm the only one with a beer in the hand. Actually, it's not true, because all the boys have, but they hide it behind the girls' back. So that's, again, thank them. And we heard it that today's conferences always, not very specialized, even a specialized one, you meet different disciplines, because the drug, vaccine, everything designed is basically a collaborative, multidisciplinary event, includes chemistry, engineering, maths, physics, medicine, ethics, and drug delivery. We are talking in the previous 2 lectures. Again, the major focus of those lectures is the delivery of the special antigen. Vaccination, obviously, vaccine conference, I shouldn't tell it, is the biggest blessing of the human race. According to different literature, increase the human life expectancy 15 to 25 years, and some diseases are completely eradicated, but none of the current medical is perfect, and vaccination included. So we have to find solution to basically create absolutely harmless vaccines, because, as you know, most vaccines are administered in early ages of the life, so the safety is extremely important. As in the previous slide, I showed that the first mass vaccination against smallpox was happened in 1798, as basically when they realized that cowpox serum actually protects against smallpox infection. It was in 1978. A little more than 10 years later, this picture appeared here, as you see that probably that was the first mass protest against vaccination, because you clearly see the fear of the effect of the different animal serums. And look at the situation in 2014. That's why it's 14, because the whole thing started. That's what we see in the today's press. And people, especially educated parents, fail to send their kids for vaccination. And obviously, this can have very, very serious consequences. I think the major reason is, I just gave an interview not long ago, is the bad press about the whole thing. Of course, there is no absolutely 100% or 1,000% safe vaccination, but the bad press is the main reason for this. Because even if it's the most successful human health intervention, problems exist. The risk of infection, autoimmune diseases, limited shelf life, especially when we're talking about third world vaccination, is a really serious problem. So the solution could be a synthetic peptide-based vaccine, as we heard in the previous 2 talks. Synthetic peptides are basically short peptide sequences, and the advantages is that remove the danger signals, so where the compounds are safe, non-infectious, customizable, but there are problems. By removing the danger signals, obviously, we don't shake the body up anymore that there is some danger, so the immune system has to start working. So need adjuvant. Adjuvant, in most cases, are toxic. Sorry, it shouldn't come in. Are toxic, so have to work on, as we saw again in the previous talk, non-toxic, generally useful adjuvants. And because of the lack of, most of the time, T helper epitope. We need some kind of T helper epitope to make and carrier to make the vaccine work in the wide population. So what is this synthetic vaccine? Here is the microorganism. Here is one protein on the surface of the microorganism, which is normally immunogenic. The immune system recognizes it. Obviously, the first step was recombinant-based protein vaccines, but it still can be dangerous because it's produced by microorganisms most of the times. So a small part of this protein is the peptide-based vaccine. If you want to have a B-cell response, you have to have a secondary structure of this. For T-cell response is not necessary. But that little thing, which basically should be administered to generate, because the antibody generates, recognizes this part, recognizes in the protein, recognizes in the microorganism, label the microorganism, and the immune system does its work. But the problem is, as we go down from the whole or attenuated microorganism to protein to peptide sequence, the immunogenicity is decreasing. And the side effect, of course, is decreasing as well. But this part, we have to do some things, have to work in the different delivery systems. So delivery system is the most important part, I think, in today's pharmaceutical, especially vaccine research, when, again, the immunogenicity increases, and the side effect hopefully won't increase with it. So many research groups in the world started to work on different delivery systems. The first one, what I really appreciated, was Jimmy Tam, who worked at the multiple antigenic system when he put lysines together. Lysine has one carboxylic acid, 2 amino function. You build a little dendrimer, and on the surface of this dendrimer or dendrimer, you put peptide sequences. This special one increases the molecular weight greatly, and the compound was immunogenic, unfortunately still needed adjuvant. A second approach was Günther Jung in Germany. It's about 25, 30 years ago, who introduced again the PAM2 cis, PAM3 cis system, which are natural bacterial cell wall lipids. And in one conference, I saw these two presentations in the same time. I thought we should put these two together. And we employed a very interesting lipid, the so-called lipoamino acid. This lipoamino acid is an alpha amino acid, has an alpha carboxylic acid amino function, can be protected amino function with different normal amino acid protecting groups, and it's extremely easy to synthesize, very cheap, a couple of dollars a kilo, or even lower than that one. You synthesize it very easily. But the most important feature of this special lipid is this is engineerable, customizable. You can increase, decrease of the length of the alkyl chain. You can dimerize or oligomerize them to increase the number of lipids. But more importantly, you can put a spacer between two lipoamino acids, so you can mimic any kind of lipidic structure what you want to build up, including the lipid A, PAM2 cis, PAM3 cis. It's extremely important because we know now that the lipids, one of the 4 building blocks in our body with peptide, proteins, carbohydrates, nucleosides, and lipids, they are not only energy providing and cell wall building materials, but they have receptors. And one of the most important receptors from our point of view are the toll-like receptors. So you have to have special structure to build up using these lipoamino acids. And we built up now several drug delivery systems. I don't go into the creation of the drug delivery systems because they are all published now, but we are using these delivery systems to find out the immune systems that I will talk about. And this system, what I introduced in many different carrier molecules, contains an adjuvant using those lipoamino acids, mimicking PAM2 cis, PAM3 cis, contains a carrier, which can be a polylysine dendrimer, can be a carbohydrate, or an oligomer of that one. When they have different carboxylic hydroxy groups, you can put different peptide sequences, and you can put one or different peptide sequences on this carrier. So the adjuvant, the carrier, and the antigen is in the same molecular entity. These systems are self-adjuvanting, hopefully, so you don't need to add any additional adjuvant. And most importantly, they are amphoteric. So then this computer just wants to pick up the signal here. Amphoteric, and it's extremely important. I'm an engineer and try to synthesize, make compounds. And I didn't really appreciate a while ago the formulation part of a work. The formulation, what these amphoteric compounds allow you to do by increasing, decreasing, or changing the lipids or the peptides, the hydrophilicity, the hydrophobicity, you can build up compounds which are have a secondary kind of nanostructure, a liposome or a micelle, or just an aggregate, what we will see these compounds can form. And I introduced the usefulness of this compound through one of the infectious diseases, the group A streptococcal infection. I'm working in a team in an NHMRC program grant, which main aim is to fight against infectious diseases, mainly tropical infectious diseases. And one of them is the group A streptococcus, which is an extremely dangerous disease in certain population, including the Australian Aboriginal population. The streptococcus infection is very common, actually. I think all of us had it, and the symptoms from the sore throat start to be dangerous when it gets into the bloodstream, invasive, and this is an extremely dangerous disease because it results about 50–60% death if you are infected once. And the biggest problem with this one is that it's highly variable, have about 200 different serovars. When the surface M protein is all different in the N-terminals, they have a relatively conserved C domain here and a relatively conserved middle domain here. And the antibody which generated against the whole protein or the microorganism is generated against this middle part as well, which recognizes the microorganisms and labels it, but unfortunately cross-reacts with the human heart, causes rheumatic fever, the consequences of several rheumatic fever, rheumatic heart disease, and death. So the fragment-based drug design is extremely important here when you use only the non-dangerous part of it. The logical is to use the relatively conserved C domain, but the antibody which is generated against it is not that protective because it's hard to recognize a peptide sequence close to the cell wall. So the combination of antibody against those sequences, which are highly variable, and the combination of variable one gives us now a structure which now gives you high protection. So we built up two systems containing four different epitopes, and the mixture of them now contains all the 7 extremely dangerous serovars from our region, and that's now is in the preclinical phase. And fortunately, because they are self-adjuvanting, they both active nasally and orally, and generate epitope-specific antibody response, and as you see, 100% protection. So this is now our first compound. First compound is in clinical 1 phase, contains only the conserved domain, but this 8 peptide sequence containing compound is in the waiting list. Is a 2nd generation. As I said, I'm a chemical engineer, but started to go to immunological conferences. And we know all those textbook materials. We know how the vaccine or vaccine particles enter to the cells. We're not 100% sure about the mechanism. We know the end result. So we have an antibody response or have a T-cell response. We can explain the mechanism. But unfortunately, all those guys who put those beautiful diagrams together and published in Nature Science, they still cannot say how to design an antigen which results in IgG response or result in IgA response, which is extremely important in the infectious diseases case, or we know that T-cell responses, but we don't know how to design an antigen which results definite T-cell response. So we started to work on this question that how to find out to design an antigen which gives you a targeted result. And we know now the size is extremely important. Again, I put this generally known graph up, which shows the different sizes, and the antigens basically, the antigens size could be from kind of 5 to 100, 200 nanometers. Those are the best ones, they say. Oops. should have switched out this kind of computer wants to find out the system. So as I mentioned, when we put those sequences together, we don't synthesize stepwise, we build the different structures, and with chemical ligation and fragment condensation, you can put large molecules together. And in generally, where we started to work now on this conserved domain from the step, and used a general helper epitope to make the compound working in an outbred population, and used a special lipid. And initially, we just put together in different configurations. It's exactly the same compound containing the helper epitope and the productive epitope and the adjuvant in different chemical conformation. And as you see, depending on the space of this exactly the same compound, you have very, very different immune response. This is a log scale, so the immune response is 100 times difference in two different structures, the same compound. So we started to examine what could the reason be. So we used this general structure again, as I said, adjuvant, carrier, and antigen in the same entity, and checked how the length of the lipid varies the immune response. And we found out that only lipids longer than 16 carbon long can generate IgA response, which is extremely important, as I said. So started to vary again now the configuration with this now C16 good lipids, and it came out that only one configuration and one special helper epitope gave you good IgA response. IgG is fine, it's generated IgA response all the time. IgA only one structure. So we realized we are lucky, so let's try out how can we generate now. Okay, we changed the helper epitope, and it still didn't give you just one helper epitope. So we started to put together the same P25 helper and this variable and conserved domain one after the other. We actually have system when eight different sequences are together. So we built this system together and checked the different immune responses. And very interestingly, when you have the variable domain in the system, gave you a much lower immune response when this variable domain was in combination with the constant domain. So this larger molecular weight compound gives you a much higher immune response. A little same situation exists in this one. The single peptide gave you a little bit lower immune response than the peptide with larger molecular weight. And as I said, these compounds are amphoteric, lipophilic, hydrophilic part. They form different nanoparticles. So again, I said the size is extremely important. The literature, again, is not straightforward. They say, well, some of them say 10 nanometer is the best, and others say 100 nanometer is the best, a micron is the best. So we measured the size of the particles they formed, and realized that the size which gave you the lower immune response was over 100 nanometer, and the ones which gave you higher immune response, it's about 10 nanometers. So check the antigen uptake of those antigen presenting cell uptake of those compounds, and it came out that the larger molecular weight compound, the larger size compound uptake is much lower than the smaller compound, and probably is not the different structure, chemical structure, but the actual size of the nanoparticle, the formulation, makes the difference. And now we are working with the system, and we be able to put it in different kind of liposomes, which we cover for oral administration. I don't have too much time to talk about it, but this system, since they are amphoteric, they can be part of the liposome building blocks, and we can cover those peptides with protective polycarbohydrate, polysugar, positively, negatively charged to make them stable until it reaches certain space in the GIT, gastrointestinal system. These compounds taken up very nicely by antigen-presenting cells give you both IgG and IgA responses. Well, thank you very much for your attention. We use this system in many other cases, and if you have any question, I'm really happy to answer in the coffee break.
I'm from Canada. Thank you very much for your lecture. Well, you're talking about immune responses, but nature provides the certain epitopes, and we don't have to design sometimes. Get the IgG or IgA response on high levels sometimes doesn't mean anything because it has to be functionally active, right? So if you have only small peptide, you can have incredible immune responses, but that immune response may not do anything for curing or preventing the disease. So I think you mentioned about there are a lot of papers published in Nature and Science and so on with good pictures, but no one knows how to design immunogen. But immunogen is provided by nature, so that we have to make sure that how we can respond to that, the immunogen that nature provides.
Well, you're absolutely right. The nature provides, but there is a big difference what form, what formulation you add that peptide sequences. I didn't talk about this whole microorganism immunization. I could, because that's really interesting how this peptide sequence base goes through a whole cycle. We went down from the full microorganism, protein, carbohydrates, peptide sequence. It goes back now. So we are using, for example, killed malaria parasite encapsulated into liposomes, whole parasite. But to answer to your question, yes, that's a big difference. How do you formulate, for example, in that group A streptococcal infection? If you administer it IP, most of the time we do it even IV, the biggest immune response is the IgG. You hardly observe any IgA response. But if you immunize it nasally in a certain formulation or orally, you get very high IgA response in a certain T helper self-help. So it depends on the formulation, and one single antigen can result, depending on the formulation and the administration, both, but in a different ratio.
Yeah, well, if I may. There is, for example, Vaxgen had the HIV vaccine on the basis of monomeric form of GP120. And they had a good immune responses, but there was no protective immune response because that you have to have a trimer of GP120 in order to generate broadly neutralizing antibodies. So you can have a good immune response, but it's functionally no use.
Absolutely. You have to have a good immune response according to, you have to have a good immune response. And that's exactly the same picture I put on. We know the mechanism exactly, but we don't know how to design an antigen which gives you an immune response and gives you the right immune response. Yeah, it has, yeah.
Okay, I'm going to take one last question, and then we have a coffee break, and that's the purpose of the small group.
We're going to do a group picture before the coffee break.
Yes, we're going to have a group picture. Thank you for reminding me.
Just a quick comment on that. So you don't need to worry, because if you want to make a vaccine or any of this, unless you can actually show protective efficacy in preclinical and clinical models, nothing is going to come out of it. So ultimately, you do have to show protection. And there are single epitopes in the case of RSV that can be completely protective, because this company is making billions of dollars just making a monoclonal antibody against that single epitope.
I agree with that. And so it depends on the system itself.
So you want to have the picture taken, and then we want to have a collective picture.